Hello, everyone. Good morning, good afternoon, and good evening, depending on where you're joining us. I'd like to welcome you to the fourth webinar in our Let's Talk MPTs discussion series. I recognize many of you who have joined so far, but for those who do not know me, I'm Bethany Young-Holt, the Director of the Initiative for MPTs in Canning Health and Morehouse at the Public Health Institute. We have over 50 people registered for this webinar today from across time zones. So as we're waiting for others to be let into the room and join, if you could please type your name, your affiliation, and where you're located into the chat feature, that would be great. We also want to note that everyone will be muted during the discussion and then will be unmuted during the Q&A. We're also recording this webinar so that we can post it and make it available. If you have any questions for our discussions during the talk, please use the chat. For any technical issues, you can chat with my colleague, Susanna Moore, who is our tech host today. Also, the recording and the related resources from this webinar are going to be available on the website, the impt.org, soon after the webinar. So today's Let's Talk MPT webinar discussion is going to provide an overview of clinical development and regulatory considerations when developing products that combine one or more active pharmaceutical ingredient, or API, for MPTs that provide HIV prevention and contraception. Today's webinar will begin with presentations focused on CMC, clinical development, and regulatory considerations with leading experts in this field, followed by Q&A. So while in 60 minutes only so much can be addressed, we aim today to highlight key issues and also to connect product developers and other leaders working across disciplines to further stimulate innovation and collaboration. A list of readings relevant to this webinar, including regulatory guidance documents, are included in the most recent webinar announcement and will be available on the website. So for those joining who may be new to the MPT field, multi-purpose prevention technologies, or MPTs, are products designed to simultaneously address HIV, other STIs, or unintended pregnancy. An array of about 24 MPTs are in development with active funding. MPT candidates include various combinations of contraception, prevention from HIV and other STIs. You can check out the MPT product database that we're actively updating at mpt101.org. And if you are aware of other MPTs that aren't included, please let us know. So we launched this Let's Talk MPTs discussion series about 10 months ago to engage experts from diverse disciplines across the globe to help address MPT and HIV prevention, R&D priorities, and gaps. Past topics include the role of cervical vaginal microbiota on contraception, HIV prevention, and MPTs, the integration of social behavioral and market research on MPT, R&D, and most recently, toxicity study considerations for MPT development. All these webinars, including ours today, will be available on the website. So today I'm really delighted to welcome our featured discussions. Kevin Hirschberger is Vice President of Integrated Drug Development at Certara and brings 20 years of experience in the pharmaceutical industry, primarily in product leadership roles, and provides integrated program management, including integrated plans, target product profiles, and financial planning. Dr. Devin Shaw is the in-house Chemistry, Manufacturing, and Control Senior Director in Certara's Strategic Consulting Division. He has more than 25 years experience in the pharmaceutical industry, including roles at Johnson & Johnson, GlaxoSmithKline, and Novin Pharmaceutical. Devin has experience working across all phases of drug development and has contributed to successful approval of multiple drugs. Dr. Tamara Goodrow is a regulatory consultant and principal at Deer Run Regulatory Consulting and has over 23 years of regulatory experience in global drug development and registration in diverse therapeutic areas. Dr. Goodrow's work includes the successful development of regulatory strategy as well as leadership in regulatory efforts, resulting in the approval of multiple drugs. And our discussion panel will be joined by Dr. Verna Schroll, who is the Senior Director of Clinical Pharmacology at Certara. Verna has over 16 years experience in pharmaceutical development, working across discovery and development phases. And throughout her professional career, Dr. Shuck's work has focused on integration of non-clinical and clinical data to drive clinical drug development strategies and regulatory submissions. So with that, I'm delighted to welcome our panelists and every one of you that's joining. And I'm going to hand it over to Kevin Hirschberger. Kevin.
Great, thank you, Bethany. Just give me 1 second here to bring up the slides, and we will put it in presentation mode. So hopefully you can see that now. Move the screen out of the way. Well, great. Well, thank you so much, Bethany. And as Bethany mentioned, we're going to be talking about today the drug development process and the development considerations for MPT, specifically clinical development, regulatory, and CMC considerations. Just a little bit about Certara. So Certara is a drug development consulting firm, enabling confident decision making throughout the entire drug development process through science and technology based approaches. Essentially, we accelerate medicines to patients, partnering with the life science innovators. Together with our partners, we advance modern drug development with biosimulations, regulatory sciences, as well as market access solutions. So just wanted to give you a quick brief on who we are and where we come from. So drug development is a long and complex process. Starting with the lead identification and optimization, profiling, and the safety and non-clinical environment, and then really moving into the phase 1 where you're going to be looking at the PK safety, tolerability, and start to look at scaling up your CMC efforts. CMC meaning that your active pharmaceutical ingredient manufacturing and your formulation development and optimization. Out of your phase 1, you will end up selecting a couple doses to take into phase 2 to study an optimal dose to take into your confirmatory safety and efficacy registration trials. So there are several influences with regards to extrapolating the efficacy and safety. Two of which are listed here: the potential for PK and PD, as well as physicochemical interactions, such as in the formulation. Also, if it's a novel formulation or route of administration. If it's, for instance, if it's a long-acting injectable, it'll depend on certain areas of the body have different PK profiles that will go. Devin will further describe MPT formulations, but here's just a quick list of influences such as the API properties, your target patient population needs as well, and the feasibility for manufacturing at scale. On the left side of the slide here are a list of some MPTs that are in development, intravaginal rings, gels, films, dissolving inserts, and so on. So MPTs are multi-indication products. It's important to note that efficacy must be demonstrated and safety characterized for each MPT. Tamara will discuss this a little bit further later on this morning, but it is also important to note that there are no regulatory precedents for MPT products. So discussions regarding your development plan, your study designs, and overall strategy is very important to have with the agencies early in development to ensure alignment and to ensure alignment with the development path. MPTs can either be a combination product or a single active ingredient product with more than one indication, as we show here. Under the combination products, there's 3 different scenarios, one of which where both agents are investigational, which meaning that there's not an approved product. The other is where you have one of the agents as an approved product or both agents as approved products. Each scenario carries with it development considerations that need to be taken into account. For instance, with regards to both agents being investigational, your safety and PK for each agent individually needs to be collected in phase 1, meaning you shouldn't jump right into phase 1 with your multi-purpose prevention technology. It needs to be characterized individually. When one agent is approved, you may have the opportunity to rely upon literature data that's available for efficacy and safety, and you may be able to bridge. But again, discussions with the agencies to make sure that there's alignment. The same goes true with when both agents are approved, leveraging safety information, and you could also possibly consider a bioequivalence approach. So for today's discussion, we'll be focusing in on vaginal rings and gels, providing the clinical development, regulatory, and CMC considerations in the area of HIV PrEP and contraception, but specifically where both agents are investigational. What's out of scope today is we're not going to go into any considerations for the non-clinical or tox program, and we won't be able to be in a position to provide product-specific or development program-specific advice. So with that, I'd like to turn it over to Dr. Devin Shaw to walk us through CMC considerations.
Thanks, Kevin. Next slide, please. So again, CMC considerations, the first 2 bullets Kevin already touched upon in his section. MPTs could be a single API or a combination of APIs. More likely, when you are targeting multiple indications, you will have to deal with combination of multiple APIs, maybe 2 or even more, because it's difficult to find a single API having efficacy in all different indications that you are targeting. And the different dosage forms also Kevin touched upon. We are focusing on rings and gels in this particular presentation because they are more advanced than other dosage forms that are being studied, and also because of limitation of time of today's webinar. I mean, there are other promising platforms, films, pods especially have shown a lot of promise. Seen examples where up to 5 APIs have been put in a pod formulation. And I guess not the concern, but the challenge is they are still in very early phases of development, and it still needs to be proven of whether they could be scaled up eventually for late phase and commercial use. But again, the focus of today's presentation is going to be rings and gels. In terms of unique challenges, especially when you are combining APIs in a formulation, every API has different physical properties: molecular weight, solubility, log-P, hydrophobicity versus hydrophilicity. So someone could be highly water soluble, whereas contraceptives typically are very lipophilic. And all these factors will determine what formulation approach you can choose that can meet needs for both APIs and arrive at that midpoint where it's optimal for formulating both the APIs. Also, different target release rates. ARVs, you typically require higher rate of release, whereas contraceptives require a much lower rate. And you need to come up with a polymer system which can achieve both of those. Stability profiles, they could have different chemical stability, they could have different thermostability. So again, in manufacturing processes, you need to make sure that they are staying stable, and the same process could be applied to multiple APIs. Surface crystallization risk, depending on API characteristics and physical form, that risk could be more prevalent for one API compared to the other, and you need to keep that in mind. Compatibility, again, related to stability profile, bullet above, equally important is compatibility between different actives, active and the polymer system you are using, and everything that will be coming in contact with during use, like condoms, tampons, and that's something that regulatory agency will expect you to show compatibility with those agents as well. And finally, alteration in local pH, again, depending on pKa of your molecule, different APIs will behave differently and lead to different levels of absorption. And you need to, again, ensure that the target rates that for both the APIs or multiple APIs that you are targeting are being achieved using the formulation that you are developing. Besides the challenges, choice of dosage form is also dependent on multiple factors which are arising from patient unmet need perspective. Product efficacy obviously is an important one, but that's not the only factor that's driving dosage form selection. Frequency of insertion, that's where intravaginal rings could have an advantage because they have longer acting efficacy compared to other dosage forms. Vaginal wetness is more applicable to gels. Insertion via applicator, which is applicable to gels versus finger application for rings. So all these different factors will feed into what dosage form is most ideal for the patient population you are targeting. And these are just this tip of the iceberg. There could be so many other factors depending on the indication and the patient population that you are targeting. And this could become very quickly quite complex. So I guess whenever developing a new therapy and selecting your indications, you have to look at a triad where you pick your indication and patient population with the unmet need. That's the first bit. Then you select which APIs will work for that indication in the patient population, and what is the best dosage form for combining those APIs. And you might have to do several permutations and combinations to arrive at the final dosage form containing the APIs, targeting the indications, which have the highest probability of success. So that requires a lot of mapping earlier on in planning, and the early formulation development work will be quite critical in determining that approach. And the final bullet on the slide, again, globally compendial excipients should ideally be used. For MPT, we understand it's a novel dosage form, novel polymers or excipients may be needed, which are not used previously. But again, where possible, you should approach the excipients where there is existing DMS provided by the suppliers so that you have less hurdle to cross from a regulatory perspective. Next slide, please. So now moving on to specific considerations for intravaginal rings. Again, APIs, as I mentioned in the previous slide, they need to show chemical compatibility between the individual APIs and with the excipients like polymers. And there could be a range of polymers, silicones, ethyl vinyl acetate, which has been used in the approved newer ring. Also thermoplastic polyurethanes, which are especially useful when you are dealing with hydrophilic molecules. Again, depending on the physical properties, you need to select the ideal polymer and need to show the compatibility between your APIs and the polymer. API should have good enough melting point, high enough melting point, so that it does not undergo any melt transition during the manufacturing process, especially hot melt extrusion, which is the most commonly used manufacturing process for this dosage form. Again, as I mentioned, hydrophilicity, lipophilicity are important because that will determine the selection of your polymers for your dosage form. DMF status of the supplier, we have already touched upon. If it's an NCE, and again, Kevin touched upon this, if it's already approved API, then probably you can leverage from the regulatory information available or literature that's available. But if it's a brand new NCE, you'll have to generate the full API package, including synthetic route, impurity characterization, stability value, and everything that's needed for EA filing for a new chemical entity. That would require a lot more work compared to a 505(b)(2) approach approved API. From a formulation perspective, again, several designs are possible for accommodating individual API properties as we went over a host of different phys-chem properties that APIs would exhibit. The simplest dosage form here is one on the left, matrix ring, where you just combine the APIs in your polymer matrix and manufacture the ring, which is delivering the release rates for both APIs which you are targeting. But a lot of times that's not possible, especially when you have large differences between the API properties. So the middle one, dual segment ring, is one example where you can have a hydrophilic API sitting in one section of the ring and a hydrophobic API sitting in another section. Again, Enovera, which is an approved product, it does belong to this dual segment ring sort of design. And then final one is the core sheath ring, again, where you are trying to control rate of your release of different APIs from different sections. And you could have one sitting in the core, the other API in the sheath, or sheath could just be serving as a rate controlling membrane. But again, that's more complex compared to simple matrix design. and new wiring belongs to that last design that we are talking about. So again, different designs could target different API properties, but would require different manufacturing considerations as well. The design should ideally be able to achieve sufficient solubility of API in polymers, because that will determine your loading dose that you can incorporate in your polymers. Target release rate is very important, because that's at the end of the day going to drive your efficacy and product performance. Adequate product stability and mechanical integrity is part of stability, but I'm highlighting that separately because especially for rings, expulsion could be a significant issue, and you need to ensure that it's hard enough so that it's staying intact during use over that month period that it might be used. At the same time, it's soft enough so that it can be inserted and it's not deleterious to the mucosal membrane in the intravaginal environment. Next slide, please. And just touching upon the manufacturing technology, there could be a host of manufacturing technologies which could go into vaginal ring manufacturing. I'm just focusing on hot melt extrusion because that's the most common technology possible and that we have come across. The diagram here is mainly depicting simple process for manufacturing, say, matrix ring, where you have a hopper, which is feeding in your API and polymers. Those are intimately mixed, and they're melted within the chamber, homogenized, and then they're extruded. And that's where for different designs, you could have different dies, so that depending on the design, you can choose the shape of the die, and that will result in pellets that you are designing from a shape perspective. And then those pellets could be combined to form the ring of your choice. But that's a very simple process, which is not usually the case, especially when you are going for the dual ring design that I was mentioning earlier, or the core sheet design. So there could be different steps depending on the design that you are targeting. There could be drug polymer compounding is obviously common starting step for every design, but then fiber co-extrusion is something that might be needed, especially when you have different layers in your final design. Cutting to size, ring welding is something that you need for any ring, but again, for dual ring design, that welding becomes important so that there's no interaction between the APIs, which are sitting in different compartments. Annealing could be required, especially for reservoir kind of design, where you want to make sure that there is equilibrium achieved for the API distribution in the polymer before they are being used, so that there's no lag time in using that reservoir ring in the patients. Rate controlling membrane, as I mentioned earlier, could be used for controlling the rate, and that would require more distinct manufacturing step in the final manufacturing process. So bottom line, the message I'm trying to convey is whether it's matrix, core sheath, or segmented ring design, even though hot melt extrusion might be the common manufacturing process, you will need distinct steps and special modifications to the manufacturing equipment to ensure that the final design you are able to achieve is scalable using the equipment and the process that you're using. And that leads to the final point about scalability, where you are typically looking at about 1,000 to 2,000 rings earlier on in phase 1, phase 2 stage. And then eventually you should be able to scale up, whether in-house or at the CDMO that you might be going with. 15 to 20,000 rings maybe at commercial scale. And you need to have that in mind. And that's the key difference between, I would say, this dosage form and more routine oral or other dosage forms, where you have more leeway of changing formulation between phase 1 and phase 3. In this case, you need to make sure that your release rates, the properties are maintained. And for that, the manufacturing process, drilling down and finalizing that earlier on is quite critical for the MPT dosage forms. Next slide, please. In terms of critical quality attributes, again, those should be guiding principles, and your manufacturing process should be targeting meeting these critical quality attributes. There are common ones. The first bullet are what you would expect for any dosage form, not just MPTs, impurities, API, assay, content uniformity, and so on. But specific to MPTs, you're looking at dimensions, the size of your tubing. The drug loading is quite important. You need to have adequate drug loading, but at the same time, not too much residual drug remaining at the end of use. In vitro drug release, very critical, as I mentioned earlier. Mechanical performance also we touched upon in terms of hardness. Composition and thickness of the tubing will drive how much drug is partitioning into your tubing and how much is being released to meet the target release rate. Sometimes particle size of API could be an important factor as well. And that there could be a host of other CQAs depending on the final dosage form and the design of IVR that you are using. Stability evaluation is equally important. A lot of times these dosage forms will be used in zone 4B. So you need to establish the stability at 3675 condition. Unless if it's impermeable packaging, then 3065 could be acceptable as well. Force degradation is also equally important, and the stability study in the in-use environment, in the vaginal pH of 4.5 to 5.5, or in contact with seminal pH, needs to be simulated and evaluated. And not just chemical stability, but the hardness and the physical stability that I mentioned earlier. A lot of times you need to make sure your ring is staying intact upon use over a prolonged period in that vaginal environment. And that you could do in a simulated fashion, either in your in vitro dissolution studies or in somewhat simulated stability studies to ensure that you are demonstrating physical stability as well. You need to have validated methods for demonstrating both physical and chemical stability, and in vitro methods are again important for both batch release, and for that you need slightly shorter method, but at times that may not be predictive of your long-term release. So you need to make sure that your method can achieve both batch release and IV-IVC if possible, using the in vitro release method. And finally, packaging is standard. You need quite robust moisture and oxygen impermeable primary packaging of multiple layers to keep these products stable upon storage. Moving on to the next slide. So now switching gears, just touching a little bit on intravaginal gels. There are two designs. The one on the left is the more recent Phexi gel, which was approved just last year. And it comes as 12 preloaded applicators. And on the right, you have more traditional, historically used design where you have a tube, a separate applicator that you apply your formulation and fill the applicator with and reuse. Ideal formulation for gel should be easy to apply, non-Newtonian shear thinning, meaning it should decrease in viscosity when you apply pressure so that you can apply it properly. Well dispersed throughout the vaginal wall, which is critical for ensuring dosage uniformity and accuracy. Residence time is a key hurdle for gels because as opposed to intravaginal rings, gels are coming in direct contact with the vaginal environment, and the viscosity is quite critical to maintain, and that will drive the residence time. And a lot of approaches are being employed as we speak for improving that residence time of gels, which I'll touch upon in the next slide. They should also be able to maintain their viscosity upon dilution in the vaginal fluid and should have good extrudability from tube and applicator in the final design. Next slide, please. And as you can see, the title, there's additional CMC considerations for gels. So quite a few things that I went over for rings from a critical quality attribute perspective will apply to gel as well. Here I'm highlighting the ones which are more critical for specifically for gel. Viscosity, as I touched upon a little bit earlier, is critical for balancing flowability, spreadability, extrudability from the applicator, and retention, most importantly. Osmolality also becomes quite important. There's WHO guidance in the range that you should be staying within, so as it's not disruptive to the epithelial membrane. Optimal pH is quite critical, again, depending on the pK of the molecule in the vaginal environment. Again, because gel is in direct contact with the vaginal content, you need to ensure that it's staying within that pH so as not to cause any irritation. And buffer capacity goes hand in hand with the pH, where you need to have enough buffer capacity in your final formulation to retain efficacy of API, both in the vaginal fluid pH and in the alkaline pH when it comes in contact with the seminal fluid. As I mentioned earlier, the biggest hurdle with gels is achieving retention at the site. And there are several approaches. I mean, I've listed separately bioadhesive and mucoadhesive, even though they are sort of interchangeable. Mucoadhesive is a subset of bioadhesive. There are different polymers used for those two approaches based on the publications that I've come across. But all these approaches are able to achieve, at least in the early phases, mucoadhesion properties. And this is not just common to MPTs. Mucoadhesion has been used for a long time in oral delivery, other dosage forms. And the same principle applies for MPTs as well, for intravaginal gels. And there could be success achieved. Right now, gels maybe are used for 2 to 3 days, but even longer term usage could be possible depending on the success achieved with these mucoadhesive dosage forms. And then there are newer technology options being evaluated as well. Thermal sensitivity gels, which are changing upon temperature change in the environment, or pH dependent release gels. And finally, in situ formation gels. All these innovation could contribute eventually to improving the residence time of gels. So again, as I mentioned earlier, CMC, this is a succinct representation of the CMC status of different MPT dosage forms. There's a lot more going on, but hopefully this provides a flavor of the CMC challenges, especially when combining APIs in your dosage form. With that, I'll hand it off to Tamara for taking you over clinical and regulatory considerations. Thank you.
Thank you, Devin. Good morning. Today, I'd like to provide you with a number of points to consider when you begin to plan for clinical development for an MPT for HIV prevention or HIV PrEP and contraception. There's a number of different scenarios that you may be contemplating, and as Kevin outlined, your MPT product might be comprised of 2 novel investigational agents, or perhaps an agent approved for treatment but not prevention of HIV, and a well-precedented hormonal contraceptive, or some combination of these. And likewise, different routes of administration may be considered, such as a vaginal ring, gel, film, subdermal implant, or oral product. For the sake of time, and just as Devin has done, I will focus primarily on considerations for development of an MPT product that contains 2 novel investigational agents that are small molecules with a vaginal route of administration. Next slide, please. Before I start, I'd like to provide some important regulatory context. As you've already heard from Kevin, there are no currently approved products for both HIV prevention and contraception. There's no regulatory precedence we can use to inform the optimal pathway for a dual indication MPT development. Further, each program and unique combination of agents, formulation, and route of administration will be different. So as the FDA, as is quoted in this slide, the FDA agrees that the development of an MPT will be complex. This is why it's critical that any organization that's wishing to develop a MPT obtain regulatory agency input early in development. And this can be achieved via the pre-IND meeting process with the FDA in the US, and the scientific advice process with the CHMP, or national regulatory authorities in Europe. Given the desire for 2 separate indications, you should make sure that you seek advice from regulatory authority experts in both contraception and antivirals. And further, given that the product, such as a ring or implant, would be considered as a drug device combination product, you may also need to take into account guidance and regulations pertaining to device development and engage the regulatory experts in this area also. Fortunately, while there's no precedence for MPT for contraception and HIV prevention, there's considerable regulatory precedence for products approved for contraception alone via a number of different routes of administration, including intravaginally. And likewise, there are some agents, Truvada, Descovy, and Dapivirine, which are approved either in the US or EU and or WHO pre-qualified for HIV PrEP or prevention. So it's important to highlight that the development program needs to meet the regulatory standards for demonstration of efficacy for each indication, and the safety of the investigational products, especially new investigational products or molecular entities, should also be well characterized. So looking at the regulatory precedents to gather insights for individual agents, to gather insights on how to develop these agents is also useful. And there's information available publicly from the Europe public assessment reports, as well as FOI information on the FDA website. So lastly, I want to highlight that there are a number of relevant FDA and EMA guidances that provide regulatory authorities thinking on how to develop these agents for contraception, HIV prevention, and also combination products. And The ones that are listed here are also provided, links are provided, I think, in the IMPT site for your background information. So these are only a few guidances that are maybe directly relevant for MPT development, but there are several other guidances that are obviously more general for aspects of clinical development, and these are certainly worth reviewing. So next slide, please. When thinking about the clinical development of an MPT or initiating clinical development composed of two different new products, before I start to discuss sort of the overall development overview, one of the key questions that has, I think just always comes up is whether you would be developing each individual product separately and then make a link or a bridge to the MPT formulation, or whether you would try to develop the product in combination. So in one scenario, each individual agent is developed independently, and then there's a bridge, usually via exposure PK, between the monotherapy formulation and the MPT. A key question here is whether, if you do develop these agents independently, a confirmatory safety and efficacy study with the MPT would also be required. Or would demonstration of bioequivalence and comparability between each monotherapy and MPT formulation actually suffice? Conversely, in the second scenario, given that each individual agent is anticipated or being tested for efficacy for a separate indication, could you use a pivotal efficacy safety data to be collected only from using the MPT without demonstrating that each agent is efficacious for its intended indication independently? So here are some considerations. For the latter scenario, how feasible is it to evaluate more than one dose in an MPT formulation? For example, if dose ranging studies are needed. The number of combinations of different doses of each product within the MPT would seem pretty challenging and would really put a big stress on your manufacturing and CMC development group. Are there PK or potential pharmacodynamic interactions anticipated or possible between the two agents? Can these be ruled out? If not, then in the first scenario, then a confirmatory efficacy safety study or studies might be required with the MPT formulation. What is the similarity between the monotherapy and the MPT formulations? Are they similar? If not, in the first scenario, then a confirmatory efficacy and safety study might be, again, required with the MPT formulation. How feasible is it to bridge from the efficacy, if you demonstrate it for each individual agent, to the MPT formulation? For topical formulations, this may be tremendously challenging, or maybe not feasible due to challenges with reproducible measurement of local exposures. Can an exposure response profile be established, and does the safety of the MPT need to be demonstrated? The answers to many of these questions really will depend upon the proposed program and the combination of investigational agents. On top of this, depending on the regulatory authority you may seek advice from, they may have different positions. So in a few minutes, I'll go through some potential scenarios, but it will be critically to think through your specific program and then seek input and concurrence on the developmental pathway from regulatory authorities. Next slide, please. So just starting with early development, and many of you, I'm sure, have some experience with this, but once you understand the pharmacology of our agents in vitro and in animals and have conducted the appropriate toxicology studies, these results will allow you to define an initial starting dose and a range for your first in-human study. At this point, you should have also chosen one or more initial formulations for testing. So generally, the primary objectives of a first-in-human or early phase studies are to collect, in this case of a topical agent, local and systemic PK data or exposure data, as well as information on safety and tolerability, including the local tolerability of your drug. During phase one, it's really critical that the PK be well characterized, especially in the case with MPT development programs, since this may be needed to help with bridging to the MPT formulation. So phase one should also allow, in the scenario we're talking about, the characterization of various rings over various timeframes to find a ring with an optimal PK profile and to assess the impact of ring-free intervals or characterization of maybe different film or film formulations. And early phase 1 studies should also allow you to gain an initial sense of the acceptability of the new formulation for the intended users and use. Phase 1 studies also allow interrogation of the MTD, the maximum tolerated dose, and in some cases may allow you to gain initial evidence of a pharmacodynamic effect. For example, with HIV prevention, your pharmacodynamics, you might be looking at the capacity of a ring to protect cervical tissue from infection upon ex vivo challenge with HIV. Capacity of vaginal fluids collected by cervicovaginal wash to inhibit HIV infection in vitro. These data plus local exposures that are measured in your phase 1 program should provide important data to help support and justify your dose selection for your key safety and efficacy studies. Other important information that should be evaluated in phase 1, in particular for MPTs, include the potential for drug-drug interactions. Are there potential PK or PD interactions between the two components of your MPT? Could local cervicovaginal changes, such as changes to the mucosa, the pH, or the flora, affect drug absorption or safety? Evaluation of DDIs with other agents that might be anticipated to be used with your MPT, such as vaginal anti-infectives, are also important to look at, as well as looking at condom compatibility. So these studies may inform whether a dose adjustment is needed and or identify prohibited co-medications. Phase 1 studies can also allow assessment of the impact of ring-free interval, the effect of menses and tampon use on PK and exposure. If you're characterizing the efficacy and safety first for individual formulations, then conduct of a bioequivalence or biocomparability study to bridge exposures between the formulations of the individual agents to the MPT formulation will also likely be important, as I'll talk about in a couple of slides. Next slide, please. So once you've collected safety and exposure data in phase 1, you may need to plan to conduct a proof of concept and phase 2 studies or phase 2 studies. The objectives of these studies are generally to demonstrate initial efficacy or proof of concept and collect data to select and justify your dose for pivotal studies. I should note, though, that to support an indication for HIV prep or prevention, proof of concept and phase 2 may be very different from a typical development program, since relatively large trials are required to demonstrate initial efficacy for this indication. Thus, development of HIV prevention might go potentially from a phase 1 to a phase 2B or phase 2-3 study. Whether proceeding from phase 1 to phase 2B or 3, it's important to be able to provide a strong justification for your dose for phase 3. So for antivirals being administered via a vaginal route, factors that might be considered when estimating the effective dose include the in vitro antiviral activity in EC50 in the relevant cell types, local exposure at different time points, and the drug release rate required to maintain the estimated efficacious exposures over the time frame of the desired duration of treatment. So exposures will need to be maintained throughout the proposed duration and will need to be measured at various time points during the treatment period. Of course, with a vaginal administration, local tolerability will also be important. The potential for development of resistance should be considered also and will need to be measured. And it's sort of noted that for HIV prevention products, higher doses may not be the most optimal doses due to higher systemic absorption if you're looking at a vaginally administered product, and the potential for adverse events or effects or risk of systemic AEs and development of resistance. Switching over to contraception, given the population at risk for contraception, it's important to identify the minimally both safe and efficacious dose. Typically, phase 2 studies for contraceptives will look at both pharmacokinetics as well as pharmacodynamic endpoints to justify a dose or doses to be studied in phase 3. However, if it turns out that you do choose to use a marketed or approved hormonal contraceptive product as part of your combination, you should be able to leverage safety and efficacy experience from the approved drug. However, the extent to which you can rely on this will depend on several factors, including comparability and exposure, formulation, and the route of administration. Still, choosing a known or marketed agent would likely help to potentially streamline your MPT development. So in the previous slide, I focused in general on the goals and objectives of phase 1 and 2 studies. I want to step back to what I mentioned earlier and provide a little more discussion around a likely potential development scenario. So next slide, please. And this slide is really complicated, I'm afraid, but it describes one development scenario for an MPT with 2 novel investigational drugs, or novel molecular entities, in which the initial development would be with the individual agents as monotherapies, and so in what I'll call the monotherapy formulation. This is opposed to a scenario where you might propose to conduct most of your development with the MPT itself. So this scenario may be preferable for initial development for a combination of 2 novel agents, since it would allow for an easier assessment of the different formulations, ring compositions, and the different doses. Also, it should allow for the identification of probable and safe and efficacious dose and release rate prior to developing the MPT formulation or finalizing the MPT formulation. Lastly, it may be preferred since it should allow for clear demonstration of efficacy and safety for each agent and for each indication, and make conduct of the studies a bit less complicated. So let's walk through this, because there's several pathways that bifurcate with this option, and you will need to decide which you may want to propose for discussion and concurrence from regulators. With this scenario, that phase 1 studies are conducted with the individual agents, and the data from these studies, together with non-clinical and in vitro data, should allow for choosing doses to be studied in phase 2 or phase 3 studies. Based on these studies, 1 or 2 doses can be chosen to study in phase 3, and for use in the development of the MPT formulation that combines the 2 agents. It's important to understand whether you would expect drug interactions, as this may require adjustment of the quantities or the amounts of API added to the MPT. At this point, as depicted in the box in the center, the phase 1 MPT DDI bridge study, it would be valuable to conduct a study to confirm whether you're able to deliver the comparable doses that you've looked at in your individual monotherapies, and determine whether there are significant PK or tolerability issues with the combination. Once you have these data, and assuming that you have data showing similar release characteristics for the MPT relative to the individual ring or gel formulations, you could conceivably decide to conduct your phase 3 trials with either the rings containing the single agent, as depicted on the sides, or potentially with the combination MPT. I didn't include arrows to that option on the slide for a number of reasons, primarily having to do with the potential timelines associated with the pathway, as well as additional challenges you might have with the pathway. Let's assume that you demonstrate safety and efficacy using the individual agent formulations in 1 or 2 phase 2 or 3 studies. I should note that if your data are compelling and you want to market either of the individual agents for HIV prevention or contraception as a single monotherapy formulation and ring, you may be able to support an NDA or marketing application for these indications at this point. But to continue with your development of MPT, you need to figure out how to bridge your efficacy data from the single agent formulation to the MPT or the combination. If your phase 1 bridging study confirms that you're obtaining comparable exposures and the release profile between the formulations is similar and acceptable, then you may consider either proposing a bridge to available data via a BE study, or simply conducting a phase 3 confirmatory study with the MPT. So note that we've been discussing vaginal topical administration, and there may be considerable challenges with demonstrating BE for a topical agent. For this reason, and also given that the combination formulation will be considered different from the monotherapy combinations, an additional phase 3 study may be required, or additional safety efficacy study may be required to confirm the safety tolerability with the combination, even when you propose to bridge data to the individual agents. So these are just a few options and pathways that might be considered or proposed. Again, it will be critical to obtain regulatory agency input on which may be acceptable in the case of your program. So I also mentioned a second scenario that would involve the development primarily with the MPT, the combined formulation, instead of the individual formulations. This approach is typically not preferred by regulators for combinations of two new investigational products. It might also be a viable option, but it may involve different challenges, both with being able to choose and test the various doses within the combination product, as well as with conduct of the studies with the MPT. And I'll discuss a few of those challenges as I talk about guidance that FDA and EMA has provided around the development of HIV PrEP and contraception. So next slide. So the next two slides discuss some of the regulatory guidance on development of drugs for HIV PrEP. And I don't want to go through I don't think we have the time to go through all the information in detail, but we'll touch on some of the questions that have come up. So per guidance, both from EMA and FDA, there's a preference for placebo-controlled trials in the case where the placebo is added to standard preventive measures, including promotion of condom use, risk counseling, and even potentially oral PrEP, which is when this is considered acceptable by local ethics review committees. However, guidance also acknowledges that following approval of a vaginal microbicide, active controlled non-inferiority trials could be appropriate. So given that there's an oral standard of care for HIV PrEP treatments, it's likely to be considered unethical to conduct placebo-controlled trials for oral agents. There's also now a vaginal ring, dapivirine, available, so it's possible that placebo-controlled trials evaluating the efficacy of this agent with the vaginal route may not be accepted. Whether placebo-controlled trials with other risk prevention measures will be acceptable will be dependent on whether the vaginal PrEP ring is registered where the studies are being conducted, how the local ethics review committees feel about this, and whether they consider it acceptable. And it's also important to get regulatory input on the question. And lastly, it may depend on whether conduct of non-inferiority trials is even possible. There are challenges associated with conduct of non-inferiority trials. Primarily, in some cases, the efficacy rates for HIV prevention agents may vary considerably between studies, and therefore the confidence interval around the positive results may be large, and definition of a non-inferiority margin may be very difficult. It may also raise concerns with interpretability of the results of a non-inferiority trial. One example we're citing is that of Descovy. The efficacy of Descovy for use in men was demonstrated with a non-inferiority trial versus Truvada. And previous trial results with Truvada in men allowed identification of a non-inferiority margin. However, efficacy results in trials for women were inconsistent, largely thought to be due to poor adherence, and thus identifying a non-inferiority margin was not possible. To support approval in women, FDA accepted a study that extrapolated efficacy based on demonstration of comparable systemic PK between men and women, and further data were requested and required on the concentration of Descovy in cervicovaginal tissues. In FDA's review, they indicated that the bridging based on systemic PK alone or based on exposure in different mucosal tissues alone was not deemed acceptable, since protective drug concentrations could differ at the mucosal levels. So ultimately, the approach was discussed during an FDA advisory committee meeting, and the drug was approved for HIV PrEP in women. The bottom section of the slide just lists the recommended endpoints for FDA and EMA. Both of them are measured via HIV seroconversion, but are measured slightly differently. One last comment is that the HIV prevention trials are relatively long. This needs to be factored in. Per the EMA, they recommend 2-year studies. FDA guidance allows for 1 year, with at least 50% of the population being followed through a duration of 24 months. Next slide, please. I'm sorry, it seems like we are running out of time. There are a number of other important considerations listed on this slide. Just to highlight, and I think most of you are aware of this, demonstration of effectiveness when you actually are conducting your phase 3 trials is influenced strongly by adherence. So that needs to be taken into account in the design of your studies. You need to take a look, at least in a subpopulation, to measure drug exposure so you can estimate the level of adherence. Tracking adherence in diary is also useful. And the frequency of the timing of administration or removal in the case of the ring relative to sexual activity is also may impact efficacy and needs to be studied. So regarding the size of the safety database, that's a question that's come up. The safety experience will consist both of safety that you've collected from any studies conducted by the individual agents, as well as safety from the MPT. Given the size of the trials for HIV PrEP, even if conducted primarily with a monotherapy formulation, that safety experience should exceed standard ICH requirements. Even if the efficacy is demonstrated and a bridging to efficacy is proposed, we would fully anticipate that regulatory authorities will require some safety with the MPT and likely require additional data that would support efficacy of the MPT. Therefore, I think the risk of not reaching a minimal numbers in the safety database is pretty low. So next slide, I'm just going to briefly talk about contraception. And my point here is I wanted, so the slide actually shows some of the key design elements, but the one thing I want to really highlight is the reason why it's very unlikely that you would be able to conduct a pivotal efficacy study for contraception and HIV PrEP in the same study. And one of the reasons is that trials that evaluate contraception are generally open label and not placebo controlled, whereas your HIV PrEP studies will be either active or placebo controlled. The trials are also expected to be from 6 to 1 year in duration. But the key primary endpoint is defined by the Pearl Index. And importantly, per the FDA guidance, cycles in which backup contraception are used cannot be included within the calculation of the primary endpoint Pearl Index. So if you look at the recommended design of the HIV prevention studies, these are placebo controlled. There's an expectation, an ethical expectation for risk reduction counseling and promotion of condom use in the HIV prevention trials. Thus, one would expect increased condom use, and you would have to throw out the cycles in which condoms were used, because this is backup contraception, which is not acceptable in the calculation of the Pearl Index to the US. The EU prefers calculation in a typical use condition, but it's unclear whether this would be considered a typical use condition if condoms were being used. So I think that the intent of this is just to point out that it likely means that 2 separate phase 3 studies for MPT, one evaluating efficacy in contraception and the other evaluating efficacy in prevention, would be needed. And there's other considerations with interpretation of data and impact on behavior also that I don't have time to get into. So last slide. So I hope that in the previous slides, I've given you sort of a sense of the complexity and the clinical development of MPTs, in particular, using 2 investigational agents with a vaginal route of administration. For the reasons I've described above, this presents one of the more challenging scenarios. Other scenarios where you use 1 or more components that allow reference to previous findings of safety or efficacy may allow you to streamline your program. I've listed several of the questions we discussed here, and the intent here is just to tee these up for your consideration as you begin to prepare for your initial discussions with regulatory authorities. I hope the discussion's been useful and will help with your planning for successful MPT development. Thank you.
Well, thank you, Kevin and Devin and Tamara. This has been such a rich discussion and so much to unpack, clearly. I know that many people have to leave. I touched base with Kevin. I don't know. We can stay on a few more minutes for question and answer. Otherwise, for those who need to hop off, just as a reminder, we will have the recording available as well as the slides very shortly on the website. So if there's a burning question, I invite you all to open up your unmute yourself and you can pose it. I know that Joe Romano had a question, so we could start off with that for anyone who can stay on. Otherwise, thank you again very much, everyone. I'd like to also just welcome Verna Schreck. She's part of the panel discussion.
So I'll throw mine out there, Bethany. My question is, how viable is it to consider any kind of comparison between the topical delivery of a drug versus the systemic delivery of a drug? Or is that something that is just not likely to be seen as appropriate from a regulatory perspective?
So I guess I'll give this a shot. If you look at the Descovy information on their approval in the FDA's position, I think it's not going to be a direct comparison. It might be able to lead you to picking a target dose if you have a systemic concentration. But I think there's a clear imperative if you start with a systemic product and exposure that you could use it as a target, but it would not be sufficient for bridging. They're two completely different routes of administration.
Right, that makes sense.
I don't know if Verna, if you have any further comments on that. She's certainly an expert in that type of clin pharm questions.
Yeah, I agree, Tamara. I think the challenge here is when you have two different route of administrations and looking at systemic versus local effects. So there isn't, you may be able to derive what the efficacious exposure targets are from a different route of administration. But, there will be questions as well in terms of local effect and local exposures. And at the end of the day, you may have to still collect information regarding the efficacy and safety for the topical, for the product. I do believe that a lot of the information that is out there are useful and helpful and can guide the drug development process. As Tamara presented, it can be very complex and complicated, and you have multiple options. So as much knowledge as you can gather for this product to help you with your decisions and your guidance, I think that's important that we do that as well.
Thanks, Verna and Tamara. We have one question in the chat. Are there any pre-IND consultation opportunities with the FDA to seek regulatory input on MPT development pathway? So if I interpret this correctly, it's even before requesting a pre-IND meeting, is there an opportunity to have a discussion with the FDA?
That's correct. That's correct. Thank you.
Great, thank you. I guess this is Tamara, right up your alley.
Right. A number of people have asked that of me before, and there's nothing officially written about that. And you probably need to talk to the FDA itself, because if you ask them if you can have more than one pre-IND meeting, you get different answers. I do think that in my experience, the antivirals division at FDA is extremely collaborative, and I think given the unmet need, it's certainly worth proposing if you feel that you need if you need specific guidance very early in development, you can certainly ask them for that. I mean, I think you could request it. I would suspect they would do their best to help you out with that.
Thank you.
Great, thank you. We have time for maybe one or two more questions, if there are any.
Hi guys, this is Bruce with FHI here. Kevin and Tamara and Devin.
Hi, Bruce.
Nice to see you guys. Quick question. So I'm very new to this space, are there any reasons why there aren't any sort, I would say, HIV PrEP / contraception pills that are already approved on the market as a combo pack? Number one. Number two, why not combine pills to I guess get to the market first, so you have a product that's available to women who need them? Those are two questions. I know, obviously, for global health purposes, we're trying to focus on more longer-term delivery systems, so there's less need for clinics and things. But are there any other reasons, other than that?
I can answer this, and others can jump in, but there is actually some work in development right now. So that's on co-formulation and co-packaging. So that's an excellent point, and that maybe Tamara, you can elaborate, or anyone.
Well, it's hard to know why there isn't any. I mean, I don't know if there's nothing in development right now on that. I think one of the advantages of maybe the vaginal administration product is it may offer a different option to women. It seems like you're correct. You could potentially take a Truvada and an oral contraceptive regimen anytime you want if you're prescribed. I think there are other user preferences that may enter into that. Is my suspicion. Yeah.
I would echo that, Tamara. There may be some cultural, working in the global health space as well.
Yeah.
There may be some cultural sensitivities to oral versus something more discreet. But that's a great question, Bruce.
Mm-hmm.
I will add else in terms of the technology, and maybe Devin can elaborate here as well, because you're looking on products that have long acting, like it's one application and you have effects that last for 3 months. I don't know of a tablet that has the potential to deliver that. So you're looking at a more frequent administration if you're looking at oral route.
No, I agree, and I think that was really the premise of my question. It's just more, is it really just a user preference? Because obviously, a daily pill that has a combination of 2 to 3 compounds that are already marketed and very inexpensive, it would be the fastest sort of route to get a product on the market. I mean, for the women, I mean, I would imagine so. Just because the development time it would be a little shorter, but anyways, I think that's good information to know.
I think there's one other consideration there, and that is the benefit risk equation for that combination product. It seems like, it may be more appropriate in areas where there's a higher risk of HIV, and there might be some concern with, really broad use if you're not in a certain situation where you're at high risk for that. So, I mean, that may be also contributing to it, because there's certainly side effects associated with that in a young women population. So the systemic pills will have a greater risk for that.
Yeah, I will add that the dual pill, it's under different names, is in development, and they are looking, NIMH and USAID are supporting some in-user research on that. And so you can look at the status of that in the MPT.101.org website, the database. But that's an excellent questions.
Yeah, perfect.
Yeah.
So there's one more question in the chat, and then Bethany, maybe we can wrap it up after that. So the question, Devin, I think this is up your alley. Could you please comment on the differences between how technical information about raw materials are disclosed to the FDA versus EMA? Secondly, have you ever seen an example of a raw material supplier being unwilling to provide adequate direct disclosure to the developer for an EMA filing? And that question comes to us from Kyle Kleinbeck.
Yeah, thanks, Kevin. Hi, Kyle. So I guess second question I can answer first. Yes, there's plenty of instances we have seen unwillingness on part of supplier to provide the information, not specifically to EMA, to any regulatory agency. A lot of times they are still in development. There could be a lot of factors driving their unwillingness, but, and that's why the point I made in the slide deck about having an active DMF, whatever they can share to lower that hurdle is critical for any new raw materials that you might be using in your formulation. I have to admit, I'm not that aware of the differences between the information required for filing to FDA versus EMA for raw materials. Again, DMF is specific for, like for Japan, there could be very different requirements than for US. And same thing for US versus EMA, FDA versus EMA, there could be differences in the information required. And that's where there are country or region specific DMFs available from the supplier to meet those requirements. But again, that's something happy to dig into and answer in more details if required.
Thank you very much.
Well, I want to thank everyone, especially the presenters and panelists for your amazingly informative presentations and also for staying on a few extra minutes for the question and answers. Thank you everyone who contributed participating in this discussion as well. So once again, we will be putting the recording and the slide deck, making that available open source online on the website. Yeah, I wish you all a wonderful rest of your day or evenings. Thank you.